thanks again to the organisers for inviting me on this trip across here, which is providing a great opportunity to spend a little bit more time in Italy and take a long awaited break actually for the year, so I'm looking forward to that. Today I'd like to tell you a little bit about the immune control of metastasis. Before I start, I did a lot of my training for 13 years at the Peter MacCallum Cancer Centre, where I was working on the development of immunotherapies and working on cancer immune surveillance. But I've had a long-standing interest in natural killer cells and particularly the immune system, and that was part of the reason I moved my group actually up to Brisbane to this new research institute, well, not a new research institute, but new research building, the central building in the Queensland Institute of Medical Research. And our group's looking for postdocs and students always to come out to Australia, and very happy to receive you and give you good training out there. So just to let you know about my disclosures there, I've got a scientific research agreement with Bristol Myers and consultancies. And the talk today, I really want to present two news stories, unpublished stories, where we've been interested in two, what we believe are novel targets in terms of cancer immunotherapy. So the first is around a new checkpoint pathway. It's not a new pathway in the sense that we've known for a long time about DNAM1 as an accessory molecule and important in the synapse between lymphocytes and target cells. But particularly our work on CD96 is what's novel. So we've published one paper on that on the knockout mice that I'll briefly touch on, but tell you more about development of a therapeutic now against that target. And in the second part of the talk, I'm going to talk about adenosine, which again is not really a new target. There's been a number of approaches to try and tackle the problem of increased adenosine in the tumour microenvironment, and this has included approaches to try and target the ECTA enzyme that makes adenosine, CD73, as well as the receptors that bind the adenosine and process that. So there's 4 receptors, and particularly today I'm going to talk to you about the A2A receptor and some very interesting data in terms of combining inhibitors of the A2A receptor with antibodies against CD73. So I thought I'd begin this talk on NK cells and metastasis by just showing you some work that we did recently with our collaborators at WeHi. We've been using this reporter mouse, the NKp46 Cree mouse, to cross into a number of different FLOX genes. And one of the most interesting ones that we worked on in the first instance was to cross into the MCL1 pro-survival factor. So a cross with NKp46 on this FLOX animal gives you an animal which has no conventional NK cells, no ILC1s and no ILC3s, as it turns out. And this mouse has been a very interesting mouse now for actually delving into the differences between conventional NK cells and ILC1s, which is really the subject of another lecture, but it's a tremendously interesting area for those studying NK cells. And this mouse really demonstrated the dramatically increased metastasis in the FLOXed animals compared to the wild-type animals. And you can see even the loss of one allele increases metastasis dramatically. And this correlates, not shown on this graph, but this correlates very tightly with the number of mature NK cells in this mouse. So this mouse has immature NK cells, which are NK1.1 positive, NKp46 negative, but it doesn't have any mature NK cells. And you can see that metastasis is widespread in the lungs, but you also see metastasis to other sites that we normally don't see in immune deficient animals. And we're using these animals now to look at early immune control of a number of different tumours, and these appear to be probably the most immunodeficient mouse we've looked at in terms of tumour control. In addition, they're a useful vehicle for transferring NK cells into and being able to understand what do NK cells need to do to control metastasis. So the molecules that I really want to talk about first in the first part of the talk is this nectin or Ig superfamily that bind nectin and nectin-like molecules. The best known of these is DNAM1, as I said, is a positive stimulus or an activator of lymphocytes, and in this talk we're really talking about NK cells. It's a member of a family that includes CRTAM as well as another activator, and these inhibitor molecules, or at least when we entered the field, TIGIT was known to be an inhibitory molecule containing an ITIM, which counterbalances DNAM. And these two molecules combine common receptors, CD155 and CD112. And 155 in particular is found on nearly all tumours that you look at. And we've got work ongoing at the moment to understand CD155, but it appears to be an important pro-tumorigenic molecule for tumours. So it's recognised by these receptors. The other receptor that recognises CD155 is CD96. And the time that we entered the field, really, the only literature that was known about this molecule was that it was a possible co-stimulatory molecule in the setting of human ADCC. So to cut a long story short, we used knockout mice that were kindly generated and provided by Marco Colonna to study these animals, and in particular we looked at these knockout mice in the context of metastasis and response to LPS. And the major findings were that the animals had an exacerbated response, the CD96 knockout mice, to LPS, and this is because their NK cells make too much interferon gamma, and they also have resistance to tumour metastasis, and also, as we showed in this paper, carcinogen-induced tumours as well. So just to show you a little data sort of representative of what we saw, you can see here the DNAM knockout mice in this model of B16 lung metastasis have more metastases than you can see in the wild-type controls, whereas the CD96 knockout mice are resistant. And the double knockout mice, which proved to be quite a useful tool in this study, always had a phenotype somewhere between the DNAM knockout mice and the wild-type animals, depending on the tumour model that we looked at. And you can see here as a reference, the TIGIT knockout mice virtually have the same sort of phenotype as wild-type animals. In another model that we've developed, a BRAF mutant, V600E mutant of melanoma in the black 6 animals, the SM1WT1 L1 line, you can see again here more metastases in the DNAM knockout animals and far fewer metastases in the CD96 knockout animals. To really try and get at where CD96 was acting in tumour settings, we've done a limited number of experiments where we've transferred in wild-type or CD96 knockout mice into lymphocyte deficient animals. So that's the RAG common gamma chain animal. Now, if you transfer this is in this case 50,000 B16 cells. These animals get a lot of metastases, so you can see 250 in the empty animal. But if you've transferred wild-type NK cells, you get a very good level of protection. But even more strikingly, if you transfer CD96 knockout NK cells, you can see these animals are essentially tumour metastasis free. I think there was one metastasis in that group of animals. And we use this kind of model, particularly in the absence of having FLOX genes, to try and prove that these molecules are important in NK cells. We then went on fairly quickly to use an antibody against CD96. The antibody is called 3.3. It's a rat IgG1 antibody. And what we did here was we used the B16 F10 lung metastasis model again. We're treating animals here on day 0 and 3, 0 being the day that you inject the tumour cells IV. And you can see in the absence of any antibody, or using control antibody, there's a high number of metastases. And then as you titrate out the effect of 3.3 from 5 micrograms across to 250 particularly, you sort of max out at this dose in terms of therapeutic efficacy. And in the subsequent experiments I'm going to show you, we've used 250 micrograms of anti-CD96. This slide's a little bit more complicated. There's 4 panels to show you here, but essentially it shows the therapeutic efficacy again of anti-CD96 in the open circles here as against the control IG, and this is in wild-type animals. And then if you, moving across to the right here, just concentrating on the effect on the anti-CD96, if you deplete all T cells, so CD4 and CD8 cells, you have a very minimal impact on the therapeutic activity of the antibody. But if you get rid of NK cells or neutralise interferon gamma, you see you completely lose the therapeutic activity of the antibody. If you do the similar experiment, this time using genetics, using a wild-type or perforin knockout animals, you can see here's the therapeutic activity of the antibody in wild types. In a perforin knockout animal, you do appear to lose some of the therapeutic activity, but it's a fairly minor effect compared to additionally losing interferon gamma neutralising interferon gamma on its own. So interferon gamma appears to be the major effector mechanism that the antibody is working through. Then if you look down here in panel C, you can see the effect again here of the antibody, but if you additionally treat the animals with anti-DNAM1, so to block DNAM at the same time as blocking CD96, you can see you completely lose the therapeutic activity of the antibody. And in this panel D here, this is really to look at the FCR dependency of the antibodies. So this is an important thing to do now that we're learning more about the importance of the isotypes of the antibodies in terms of therapeutic activity. As I mentioned to you, this is a IgG1 antibody. We weren't expecting it to work through activating FCRs, and indeed that's what we saw. So you can see therapeutic activity in each case in the Fc gamma R3 knockouts or Fc gamma R4 knockouts, or even in this case where there are more metastases basally in the complete activating FCR deficient animals. Again, there's still therapeutic benefit in these animals. We then wanted to look at mechanism and control in the context of other checkpoints. So firstly, just to compare the therapeutic activity of anti-CD96 alone with the well-known checkpoints anti-CTLA-4 and anti-PD-1. Now these antibodies have typically been used in primary tumour settings rather than metastasis settings, and you can see that they have fairly minimal impact alone in this metastasis assay, in this case B16F10 here, and over here RM1 prostate carcinoma. And interestingly, anti-CD96 you can see has a superior therapeutic activity, but particularly when used in combination with anti-PD-1, there's a very nice combinatorial effect of the two checkpoints blocked together. And this same pattern is also seen in the model of RM1. If we delay treatment out to day 5, which doesn't sound very much, but in B16F10 that's a substantial amount of established lung colonies at day 5, we can still see some effect of anti-CD96, again minimal effect of anti-PD-1, and an improvement in terms of metastatic control with the combination of anti-PD-1 and anti-CD96. We have gone on and looked at this combination, particularly in terms of mechanism, more recently, and so this slide's just to show you that the number of NK cells per lung is elevated with the combination with anti-CD96 and anti-PD-1. The number of NK cell, proportion of NK cells, I should say, making interferon gamma is also elevated in the combined therapy group, and we see an increased amount of interleukin-2 and interferon gamma coming from the mononuclear cells in the lung with this particular combination again, and this, at least in part, I think speaks to the therapeutic activity of this combination. We've also looked at beyond experimental metastasis, we've looked at spontaneous metastasis. So in this setting we can use the E0771 mammary tumour. So this is a tumour that you can inject into the mammary gland orthotopically, then commit to surgery, and in this case 16 days later doing surgery, and then treat the animals with a course of therapy, as you can see here, control IG or anti-CD96. And so this is treatment of true spontaneous metastasis post-surgery, and you can see control here of by anti-CD96 is quite impressive. These are the negative controls, if you like, with the CD96 knockout animals, and here's the TIGIT knockout as a comparator. So you can see that TIGIT loss in terms of the knockout is pretty much has a relatively null effect, but particularly in combination with anti-CD96 we see very low levels of metastasis. We've also had a chance to look at, we're doing more of these experiments at the moment, but looking at blocking TIGIT and anti-CD96 in combination. And so this is another example, this time in B16F10, using the TIGIT knockout animals and combining that with anti-CD96. You can see a very nice combinatorial effect of blocking these two pathways. And this is also seen in the RM1 model again, quite strikingly here. So we believe co-blockade of these two pathways can be a very effective new strategy for suppressing metastasis. So in terms of the future work, I've really just touched on the mouse work that we've been doing, and I've talked really exclusively about metastasis in this presentation, but we're also working on primary tumour growth, established tumours and treatment of those. And CD96 is expressed on, basically in the mouse, is expressed on all lymphocytes. It's high and constitutive on NK cells, but it's also found on CD4 and CD8 cells, and particularly is elevated after activation. And so we have plenty of evidence that that antibody works against primary tumours as well. So we're developing this target now as a therapeutic. We've got a program where we're making humanised antibodies. This is a partnership with Bristol Myers Squibb, and we're obviously generating these antibodies and characterising these to look at enhanced lymphocyte function by blocking CD96. There's a lot of extra work to do in the animals now. We're moving into humanised systems to test the anti-human antibodies, but we're also doing a lot of work characterising the expression of the receptors and ligands in different mouse and human systems. I didn't talk much about safety, but we haven't seen any adverse effects of co-blocking TIGIT and CD96 or PD-1 and CD96 together, and we've actually generated double knockout animals now as well and evaluating those, and thus far these animals look relatively normal. And of course we want to evaluate the function of CD96 on other lymphocyte subsets. So I mentioned that we have begun to do that on T cells now, but we're also creating a conditional allele of CD96 so that we can study this in a more precise manner. So the second part of the talk, I really wanted to talk about adenosine, and this is a cartoon to really just show you that increased levels of extracellular adenosine in the intratumour environment is immunosuppressive, and this is driven really by hypoxia, HIF1 being one of the major upstream factors controlling CD73 expression and adenosine generation. And the biochemical pathway here is that ATP is converted into AMP by an enzyme called CD39. The AMP is then turned into adenosine by the ECTA enzyme CD73. And adenosine can bind these various receptors, particularly the ones of interest on immune cells are the A2A receptor and the A2B receptor. And so our group's been actively studying those. The adenosine has multiple effects on different cell types. In the interests of this talk, really, we've been mostly concentrating on the effects of adenosine on lymphocytes, particularly effector T cells and NK cells. The published literature suggests that adenosine can suppress the cytokine production by NK cells, the maturation of NK cells, which is something that we're actively testing at the moment. It can also affect the effector molecules and the granule exocytosis process that NK cells use very effectively, as well as a whole range of other effects. So it's a complex system with a number of different potential points of mechanism of action. It's actually quite a challenging pathway to study mechanistically. So one of the important points about the A2A receptor is that it's quite highly expressed on NK cells. Unfortunately, of these being GPCRs, there hasn't been really any successful production of antibodies against either the A2A receptor or A2B receptor, and that's a bit of a stumbling point for the field at the moment. But we've used RT-PCR to basically characterise levels of expression. You can see amongst the lymphocytes, NK cells really express quite a lot of this receptor, and that really pricked our interest. In terms of the phenotypes, the A2A receptor knockout has a very strong phenotype for suppression of metastasis. So this is really shown here in the context of the B16F10 tumour overexpressing CD73. So these are CD73 high cells, and you can see the dramatic suppression of metastasis. One can also treat animals with the A2A receptor inhibitor. This is the Schering-Plough compound that we've used here. There's a number of these compounds that have been generated, both tool compounds and compounds for human clinical use. So these have been used in Parkinson's disease by a number of companies in trials and reached basically phase 3 with compelling safety profiles. So this is a drug which is very interesting in terms of repurposing really for oncology. So you can see the effect of the A2A receptor inhibitor here is quite impressive in terms of suppression of metastasis. I should say that these inhibitors have a very short half-life, just in the range of hours, and their activity against primary tumours is actually a lot more limited in terms of activity. So probably generating antibodies against this receptor could be a good strategy, I think, for probably improving the effect against primary tumours. Interestingly, if you combine the A2A receptor inhibitor, the 58261 compound, you can see here, here's the activity of it alone in the B16 CD73 high tumour, so you can see quite a dramatic activity, but greatly enhanced activity in the presence of anti-PD-1, and you can also see an improvement with anti-CTLA-4 or anti-TIM3 as well. Notably on the right side, you can see here that the A2A receptor inhibitors really have very little therapeutic activity in a setting where there isn't a lot of CD73 being generated. So in this context, there are CD73 positive cells in the microenvironment. Some of the immune cells express it, but really tumours that express a lot of CD73 are probably going to be the preferred target group. And for particular types of cancer like triple-negative breast cancer, there is a proportion of patients that are CD73 high, and they do very, very poorly. We showed that in a very large study of breast cancer patients. In addition, we've been able to look at the efficacy of the A2A receptor inhibitor and the anti-PD-1 in the context of T and NK cells. So this basically just shows the effect of the combination here, and you can see here by depleting NK cells you completely lose therapeutic activity. In this case, there is a fairly significant effect though of T cells as well, so if you deplete either CD8 T cells alone or CD4 and CD8 T cells together, you also lose therapeutic activity. And this just shows you what happens to the NK cells in the context of anti-PD-1 and the A2A receptor inhibitor. So an increase in the proportion of NK cells and NK cell numbers in that particular setting in the lungs. So does inhibiting adenosine generation and signalling display redundancy or improve anti-tumour activity? This was a question we thought that there would be redundancy in these in this system potentially. So we've looked at combinations of the A2A receptor inhibitor with anti-CD73, and you can see that there's a tremendous benefit in combining these two agents together over the single agents alone. And that's also shown in the spontaneous model of E0771 lung metastases here. We've got very few metastases with the combination compared to the single control. In addition, we've looked at trying to target CD73 with an enzymatic inhibitor, APCP, with the A2A receptor inhibitor, and we do not see that synergy. So clearly, CD73 is doing something independently of its enzymatic activity in terms of its ability to combine with A2A receptor inhibitor in these models. We also looked at the mechanism in the context of this combination, in the context of knockouts of subsets of cells. You can see again it's NK dependent and independent of the FC receptors in the case of this combination compared to the controls. We've also looked at survival in the context of the 4T1 model. So this is again a surgery and treatment metastasis model. And you can see the combination of the A2A receptor inhibitor with anti-CD73 is superior over the monotherapies. And if we look at this mechanistically, the combination activity is lost if we deplete T cells or NK cells. Either of those appear to be important in the mechanism, and particularly interferon gamma reduces the therapeutic activity of this combination in this 4T1 model. And finally, just to show you in terms of primary tumour formation, we've looked at the double knockout mice, the A2A receptor CD73 double knockout animals, and you can see that in this case this is the SM1WT1 BRAF mutant melanoma, and we can see very good suppression. This is quite a difficult tumour to treat actually with immunotherapy, but by knocking out both A2A receptor and CD73 we get very good suppression. And this is also seen if you measure tumour mass at endpoint. So in summary, improved anti-tumour immune responses in the A2A CD73 double knockout mice, which is interesting. I don't think that would have necessarily been predicted at first. Combination therapeutic strategies via the adenosine receptor antagonism and CD73 can improve metastatic control. And it really highlights the potential and the potent immunosuppressive functions of adenosine within the oncology setting. So I'd just like to finish there, thank the people involved in the work, particularly the study with CD96. I'd like to thank Ludwig Martinet here, this French postdoc who's returned back home now, but he initiated really the CD96 studies and published the Nature Immunology paper I showed you, as well as contributing a lot to the therapeutic development of CD96. Others involved in that particularly have been Kim Stannard and in Michelle Teng's group, Stephen Blake, and I'd like to thank them for their contributions, as well as John Miles. And for the work on adenosine, particularly like to acknowledge Arabella Young, a PhD student in the lab, Deepak Mittal, and long-term collaborator John Stagg, and of course the people who funded the work. So thanks very much.